My name's Dan Wozniak. I'm part of one of the newest basic science departments here at OSU in the College of Medicine, Microbial Infection and Immunity. And as the name implies, it's kind of a microbiology and immunology department in the med school. I also have a cross appointment in College of Arts and Sciences. I'm in the microbiology department there. And all our labs are located on the 7th floor of the Biomedical Research Tower in the Center for Microbial Interface Biology. And so, today, what I just thought I would do is give you a very brief overview of kind of what goes on in our group. And I also oversee a big group of investigators here at OSU that study bacterial biofilms and their intersection with both the host and the environment. And it's a rather large interest group that we have here. So my lab is primarily interested in opportunistic pathogens, and as the name implies, these organisms don't cause disease in healthy individuals. So, if you have a healthy immune system, it's really not a problem. But these organisms can cause diseases in certain patient populations that are immune compromised. And the organisms that we study in the lab really focus primarily on three: Pseudomonas aeruginosa, Staphylococcus aureus, and the bacteria Acinetobacter baumannii. And again, all of these are organisms that don't typically cause disease in healthy individuals. You have to have some kind of underlying complication. And most of my work since I've been here has focused with Pseudomonas, but what we learn in this organism, we try to apply to these other important pathogens. So just a few background points on what the types of diseases these organisms cause. They're shown here. We typically break them down into acute and chronic infections, and the list of acute infections is shown here. Things like urinary tract infections, ventilator-associated pneumonia, sort of opportunistic hospital-acquired infections. And chronic infections like pneumonia, chronic airway infections in people with cystic fibrosis. Two of these organisms are really important there, and chronic urinary tract infections, and all three of those organisms we just talked about are real major players in wound infections. And so, as you can see, just looking at the types of diseases here, all these individuals have some kind of underlying complication, whether it be in their sort of innate immune system, whether they're catheterized, whether they have some invasive procedure, their healthy immune system is disrupted, and that's how these organisms can get in and cause a foothold and cause disease. Okay, and so the two types of infections that we talked about, the chronic and the acute infections, can also be broken down into sort of how they cause disease. And really simplistically, the acute infections oftentimes disseminate. They enter into the body but disseminate, and they invade. And these organisms are typically called planktonic bacteria. So they're free-swimming, and they invade tissues, and they can cause a very acute illness. But they're also much more susceptible to treatment. And but on the other hand, there are the chronic type of infections that we talked about. These are localized. They rarely spread outside of the organ of which they're infecting. But these organisms, because they're growing in these complex communities called biofilms, are also very less susceptible to treatment. That includes antimicrobial therapies that we throw at them, and also the host defenses that the body has in order to try to rid these infections. Okay? And again, all of these organisms that we talked about have the capacity to cause either of these types of infections. And so I'm not going to go over any data today. If you want to read it, we can talk one-on-one about data if you're interested, or you can read our papers. The last slide I have in this slide set has some of our recent publications, but I just want to touch on the funded projects in the laboratory, and they're shown over here. So we have one project that deals with just sort of the genetic regulation of how this switch between acute and chronic infection occurs in the host environment. And particularly that's relevant in the disease cystic fibrosis. Again, this is a genetic disease, one of the most common genetic diseases among Caucasians, and virtually everyone with cystic fibrosis gets pulmonary infections, and most of them unfortunately die from those chronic infections in these individuals. So it's a major health problem in the United States and abroad. We also are very interested in how those biofilm communities are put together and assembled. And we have a basic science grant that really looks at the interplay of how these biofilms are assembled, and particularly the role of certain polysaccharides in holding them together. And we're also interested very much in how that structure imparts the resistance properties of the biofilm. And so we're doing studies with the interface of innate immune cells, primarily macrophages and neutrophils, and how they see biofilms and how they deal with biofilms when they're presented in that host environment. We also have animal models for all of these infections. We have acute models, primarily the mouse models, but we also have recently developed, in collaboration with Shannon Sen's lab here at Ohio State, a chronic wound model where we're able to infect wounds with these organisms and study the course of wound colonization over time with each of these pathogens in monoculture, but also in co-cultures with each other, because many of these infections in the actual host environment are polymicrobial. They're not just one organism. They're usually these primary organisms plus some other ones even. So we have a funded collaborative project to work at that. And finally, we're also we have some pharmaceutical funding and a recent R41 grant that looks at therapeutics to try to target these infections. So, as I said, the therapies for acute infections they're reasonably well, apart from antimicrobial resistance properties, they're reasonably well managed, but these chronic infections are very difficult to treat. And with our knowledge, basic science knowledge of sort of how the biofilms are structured, we're figuring out ways that we can target monoclonal antibody therapies either alone or in combination with traditional antibiotics to get in there to target these biofilm communities and break them down so that more reasonable strategies can be used. And so we're working with both pharmaceutical companies and some NIH funding on that. And then finally, during the course of chronic disease, these organisms are there for literally for years and even decades in these chronic infections, and they undergo adaptations. They change their sort of phenotypes over time, and this is a really interesting area to look at sort of how bacteria evolve over time in a real-world situation in a host environment. And so because our chronic infections last for long periods of time, we're able to sample the population at various time points and do whole genome sequencing and look at how the bugs are adapting over time to these inflammatory environments. And so we have a funded projects to work at that, to look at that also. So again, no data, just sort of giving you an overview of where we're at. If you're interested in, here's some recent publications from our laboratory, and I'm happy to talk to you on a one-on-one basis if you want. If any of this sounds of interest to you, you can set up a one-on-one appointment, and I'm happy to talk to you individually if that's okay.